Good morning. It's December 8th, and this is your daily brief in science. Here's everything you need to know. A new data set from ASH 2025 sheds light on SLS009 combined with azacitidine and venetoclax for relapsed or refractory AML with prior venetoclax exposure. In 35 evaluable patients, the regimen produced a 46% overall response rate in this VEN-pretreated group. The least pretreated cohort showed a median overall survival of 8.9 months, well above a historical benchmark near 2.5 months, while those with one prior therapy had not yet reached median overall survival. In that one prior therapy group, the overall response rate reached 58%, and the median overall survival was not reached, suggesting durable responses in less heavily pretreated patients. Across all cohorts, median overall survival exceeded historical benchmarks, with the strongest signal in the least pretreated group. The trial enrolled a median patient age of 69, with 98% of patients in ELN adverse risk, and frequent mutations in ASXL1, RUNX1, TP53, and SRSF2. SELLAS notes the data support its broader GPS program, and SLS009's aim to counter resistance to venetoclax-based regimens, including activity in ASXL1 and TP53 mutant AML-MR plans, call for expanding the study into newly diagnosed high-risk AML starting in Q1 2026 to evaluate SLS009 plus AZA/VEN in that setting. In mutation subgroups, ASXL1 mutant patients showed a 48% response rate, including 19% CR/CRY, and TP53 mutant patients showed a 57% response rate, including 29% CR/CRI. The SLS009 plus AZA/VEN regimen at 30 mg. IV twice weekly was well tolerated with no dose-limiting toxicities or treatment-related deaths. Lead author Dr. Joshua F. Zeidner presented the ASH 2025 poster in session 616 at the Orange County Convention Center in Orlando on December 7th. The release notes include forward-looking statements and investor contact information as part of the disclosure. AOP Health unveiled new clinical results at the ASH annual meeting in Orlando, spotlighting 2 Ropeginterferon alpha-2b studies in myeloproliferative neoplasms to advance treatment strategies. The presentations focused on essential thrombocythemia and polycythemia vera, underscoring Ropeginterferon alpha-2b's potential role in these conditions. Ropeginterferon alpha-2b is the first interferon approved for polycythemia vera in the EU, delivered as a long-acting monopegylated formulation with a dosing schedule starting bi-weekly and potentially extending to every 4 weeks after stabilization. Background notes describe Ropeginterferon alpha-2b as a proline interferon with a long-acting monopegylated profile and detail its regulatory status and administration. In the EU, Ropeginterferon alpha-2b is used as monotherapy for adults with polycythemia vera without symptomatic splenomegaly, administered via subcutaneous self-injection every 2 weeks, potentially extending to 4 weeks after stabilization. The ROP-ET study is a prospective, multicenter, single-arm Phase 3 trial evaluating Ropeginterferon alpha-2b for essential thrombocythemia patients who cannot receive standard cytoreductive therapies, focusing on safety and efficacy. ROP-ET targets patients with essential thrombocythemia lacking suitable conventional treatments, assessing Ropeginterferon alpha-2b's safety and efficacy in this difficult-to-treat group. AOP Health emphasizes a commitment to addressing unmet patient needs in rare diseases through ongoing research and development, aligning with the ASH presentation. The company highlights its Austrian roots, global reach, and philosophy: need, science, trust, as it focuses on rare diseases and intensive care medicine. Context: AOP Health is a multinational group headquartered in Austria, working with subsidiaries and partners to advance research, physician collaboration, and patient advocacy in rare diseases. The BESREMI-PASS study provides real-world evidence of Ropeginterferon alpha-2b in polycythemia vera, offering practical insights into everyday clinical practice. BESREMI-PASS analyzes Ropeginterferon alpha-2b performance outside controlled trials, highlighting real-world treatment outcomes for polycythemia vera patients. CEO Martin Steinhardt reiterates that AOP Health aims to share new findings with the scientific community to advance treatment strategies for chronic blood cancers. A Japanese team revisits Lord Kelvin's knot concept with a modern twist, proposing that knotted field configurations naturally arise in a particle physics model that extends the standard model with gauged B-L symmetry and Peche-Quinn symmetry. They publish in Physical Review letters, linking knot structures to neutrino masses, dark matter, and the strong CP problem. As the universe evolves, a knot-dominated era gives way to radiation domination, and knot collapse emits particles that reheat the cosmos to about 100 GeV, tying the mechanism to the epoch when neutrino-induced asymmetries could produce more matter than antimatter. Gravitational waves arise from knot dynamics, offering a potential test via future observatories such as LISA, Cosmic Explorer, and DECIGO. Observational prospects include LISA, detecting signatures of a knot-dominated era through the gravitational wave spectrum. Early universe phase transitions could generate cosmic strings whose interactions stabilize knot solitons, which then dominate energy density before decaying through quantum tunneling. Knot decay yields heavy right-handed neutrinos, driving the observed matter-antimatter imbalance. In this framework, stable knot solitons form in the early universe and unravel via quantum tunneling, releasing heavy right-handed neutrinos that decay with a slight matter over antimatter preference, producing the baryon asymmetry we observe. The work is led by Professor Munetto Nitta with colleagues Minoru Ito and Yu Hamada, proposing that knots formed during phase transitions influenced baryogenesis. The study presents a testable revival of Kelvin's idea, aiming to connect knot dynamics with observable cosmological signals, including a reheating temperature near 100 GeV and a gravitational wave background altered by neutrino-induced asymmetries. The model keeps Peche-Quinn symmetry global to preserve axion-related dark matter solutions, while gauging B-L to ensure heavy right-handed neutrinos and knot stability through Chern-Simons coupling between PQ vortices and B-L flux tubes, offering a concrete link between knot physics, axions, and neutrino mass generation. Linozyvic is being positioned as a potential frontline therapy or monotherapy for newly diagnosed multiple myeloma, with multiple linker studies exploring its use across transplant-eligible and ineligible patients. In the linker MM6 trial, induction with DRD is followed by linozyvic monotherapy versus continued DRD for transplant-ineligible NDMM, and several other linker studies span various phases and indications. Phase 1-2 data from linker MM4 show encouraging linozyvic monotherapy results in both transplant-eligible and ineligible adults. Safety signals include mostly grade 1 cytokine release syndrome and neutropenia as common treatment-emergent adverse events, with infections occurring in a large portion of patients but rarely at grade 4 or higher. Among MRD-evaluable VGPR-positive patients, 95% achieved MRD negativity at 10 to the -5 sensitivity, with 19 of 20 evaluable VGPR-positive patients across all dose groups achieving MRD negativity at this level. The REMS program remains in place due to CRS and neurologic risks, with step-up dosing and inpatient monitoring after initial doses and ongoing infection, liver, and hematologic monitoring. Post-induction, 10 patients proceeded to autologous stem cell transplant with adequate CD34 positive yield, demonstrating feasibility of sequencing transplant in the linozyvic-treated NDMM cohort. In dose escalation and expansion phases, 45 patients showed a median time to response of about 1.2 months, with CRS and neutropenia as the most common TEAES and no grade 4 and higher infections or dose-limiting toxicities. Across dose groups, LD monotherapy results show VGPR-positive rates of at least 70% in NDMM, with expectations of deeper responses over time. Linozyvic is a BCMA x CD3 bispecific antibody built on Velis Immune technology, designed to redirect T cells to myeloma cells, with ongoing development across earlier lines and precursor conditions. And the broader program includes Velis Immune technology and REMS safety measures across linker trials. Regeneron emphasizes that linvoseltamab is investigational and has not yet been evaluated by regulatory authorities for frontline NDMM, with ongoing safety and efficacy data forthcoming. A Harvard-backed study traces gut-derived metabolites all the way to the liver and beyond, linking specific molecules to metabolic effects. The researchers map how these metabolites travel from the gut through the hepatic portal vein to the liver and then into the bloodstream, influencing hepatic metabolism and systemic insulin response. The liver acts as a central hub, receiving signals from gut microbes and distributing them to the body, offering a potential new angle for treating obesity and type 2 diabetes. Published in Cell Metabolism on September 5, 2025 by Munoz et al., the work was funded by the Sao Paulo Research Foundation. Modifying the gut microbiome with antibiotics altered metabolite distribution between portal and peripheral blood, confirming the microbiome's role in metabolite production. The findings point toward therapies that mimic or promote protective microbiome-derived metabolites to reprogram metabolism for metabolic disease management. Future therapies could target microbiome-derived chemical signals to rewire fat and glucose handling, rather than focusing solely on appetite control or blood sugar. Mesakinate, a Krebs cycle-related metabolite, increased after antibiotic treatment and improved insulin signaling when applied to hepatocytes, suggesting therapeutic potential. Specific gut-derived metabolites like mesakinate enhanced insulin signaling and modulated fat storage in liver cells, demonstrating a direct gut-liver metabolism pathway suitable for targeting. In healthy mice, the portal vein carried more than 110 distinct gut-derived metabolites, while obese and diabetes-prone mice on a high-fat diet showed a marked reduction in enriched metabolites reaching the liver. Genetics and environment shape metabolite profiles, with differences observed between obesity/diabetes-prone and metabolically resilient mice, and on a high-fat diet, portal vein-enriched metabolites dropped from 111 to 48 in obese-prone mice, highlighting how diet and genetics influence which microbial products reach the liver. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.